Okay, then welcome everybody. We might just get started with the introduction. We could look at the next slide, please. Okay, so today's QAAFI seminar is by Alberto Baldelli. He'll be talking about the application of particle engineering in food products. And I'm the facilitator, Dr. Bernadine Flanagan. I'm a research fellow in the Centre for Nutrition and Food Science here at QAAFI at UQ. I'd like to give an acknowledgement of country. So the University of Queensland acknowledges the traditional owners and their custodianship on the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. Thanks, Alberto. Okay, so just some housekeeping. This seminar is scheduled to run from 12 to 1. We will aim to finish by 1. Throughout the seminar, you can ask questions using the Q&A function. Please don't use the chat function. We'll be keeping questions to the end, and I'll relay them to Alberto for him to answer. So some background on Alberto. Alberto is a fresh face to UQ. He started here this year. He is a special joint appointment, but as a lecturer in SAFS and a researcher in the Centre for Nutrition and Food Science part of QAAFI. He graduated with his PhD in 2016 from the University of Alberta in Edmonton in Canada. And following that, he worked as a postdoctoral research fellow at the University of British Columbia. And then he worked at a pharmaceutical company called Hovione. His areas of research are the encapsulation of bioactive compounds, spray drying, edible spray coatings, particle formation, evaporating droplets, inhalable drugs, and powder engineering. All sounds very fascinating. To date, he's published over 60 research articles, 3 book chapters, and produced 5 patents. So today we're going to hear about his work he's done in the past and also what he will continue to investigate while he's here at UQ. So I'd like to hand it over to Alberto.
Hello, welcome everyone. Thank you, Bernadine, for your introduction. Nice to hear somebody else that talks about myself. So what I want to do today is raise awareness of particle engineering, which is not really known worldwide, and how you can apply that to food engineering. Bernadine already introduced myself, so I just want to summarize a bit. I did everything, all my studies and research fellowships in Canada before the University of Alberta, which many people talk Alberta and Alberta saying the same. It's actually a cool university in the northwest cities in Canada, in Edmonton. Very cold, very different than what you are used to here. And that was on particle engineering. My PhD was funded by AstraZeneca. Particle engineering is a theory that is applicable a lot for both pharmaceutical products and food products. After that, I moved to University of British Columbia, where I did research fellowship and postdoctoral fellowships in the Department of Food and Land Systems. So that's where I start applying what I knew onto food. Before I continue to discuss what particle engineering is, I would like also to acknowledge the traditional owners and their custodianship of the lands of which we meet today, and pay my respects to their ancestors and their descendants. So what's going to happen today? So this is the bullet points of what I'm going to talk about. So particle engineering is the theory that deals with powders. So I'll start introducing powders, how powders are applicable to food. And then I discuss what is particle engineering. So what it actually is. So the theory, so I'll give you a little bit brief intro of the theory and what is actually teaching right now in food engineering too, here in ECFAS. Later on, I discuss what I've done before. So before coming over here, which has happened about 5 months ago. And what I intend to do here to you, I'll give you some brief examples of what I envision myself working on. These are some examples of powders you all see and you all deal with them in your daily life. When you go to the grocery, you use flour to make bread and to make other bakery products. You can also use almond flour if you are gluten allergic or for having healthier products. Powder is also including salts and all the condiments like spices, turmerics, or peppers, black pepper, paprika, all of them are powders and can define me as such. Other things that are less common is food colorant. You see at the bottom center, beautiful colors obtained chemically, not very friendly for our body. We also have ready products like cakes. Those are all powders. And then we have dairy powders, which is extremely high down here in Australia and even more in New Zealand. So an example here, I put like a dairy powder, or it can be also lactose powder, can be also whey protein if you do some sports. Ready ice creams, a very interesting product, which is sold as powder. And also this Milo is a very Australian supplement products, also made of powder. Why these powders are so great and so popular in food is because their shelf life is at least twice longer than the fresh food, and in some cases it's reaching even 10 times. So if you consider milk when you buy from your grocery and you put it in your fridge, it's not going to last longer than 1 week at most. And the powder, however, is going to sit in your shelf for over 6 months and even more in some cases. So that's the main difference. And it might make a difference for yourself, for your daily life. So you can have some powder at home. And if you forget to buy the milk, you can use it and disperse in water. It's not the same pleasure that you might get from the fresh milk. I do agree. But for the company perspective, this is an increasingly crazy high amount of revenue. So when you think about a dairy company, when they produce milk, and they cannot sell all of the fresh milk they collect daily. So what they do, most of them, is creating powder, but they store it and then redisperse in the water and sell it as a fresh milk, rather it be in Australia or outside. New Zealand is very famous for that, and that's what has been doing for about now 20 years. So that's the main interest that drives particle engineering into food. Some example here is what's happening economically. I talk about some revenue. You see on the left-hand side, US, which is still the largest economist in the world. It's obviously now topping up with China. Both of them are buying and producing increasingly crazy amount of powders in general. What you see on the left-hand side plot, you have a trend, what happened in 2020 and 21, and the forecast is what's going to happen until 2030. The number that you see is 1.7, 1.8 billions is what the market of powder in food is. On the right-hand side, you have a niche market, a smaller one, Australian. Obviously, we are way smaller in population, so the market is obviously way smaller than what US or China is. But the example down here is egg powder, which Australia is one of the highest produced in the world. You might not have heard about egg powder, and it's funny because sometimes when you go to the grocery here, you don't even find the eggs. So a supplement that people do and buy, it's powder. So you get the powder at home. You can put directly powder in your pan with a little bit of water or oil or butter, whatever you prefer, and you create scrambled eggs. You can create omelets, you can create all the products of bakery, cakes, all the products containing egg and itself. So the egg powder market from Australia is in US billion dollars, so it's quite high. We envision, so last year in 2022, we have 1.1. This year is 1.2 billion of egg powder sold in the world from Australia, and we see that increasing for future. So there are lots of industry interested in self-powder particle engineering and how to improve their powder, the properties, and how to increase their yield in production. The powders that I mentioned before and the ones that are part of these large markets are produced by different type of devices. You see here the two main types. One is the spraying techniques, which I'm expert in and what we're going to talk today. So the one you see in the blue circle is all of the spraying world, spray drying, fluidized bed, roller mills, and all the mills things. And on the right-hand side, you see it's a simpler version, so the size reduction theory. So this is when you collect flour and you want to crush it down into smaller size. Usually smaller sizes are helping powder to redisperse first better in water and to have a low spacing for transport. So those two are working together, or sometimes one or the other. I just want to show them to all of you to be aware of what I'm going to talk today. So what I'm going to discuss today is mostly on the left-hand side. The other side is also there, and it's equally valid. So the particle engineering theory, it's all revolving around a number called Peclet number, which is at the center top in this slide. It's labeled as PE, and that's equal to the ratio between kappa. Kappa is the evaporation rate, which is defined in the little sketch at the top of the slide. You see the yellow arrow drawing into the droplets. So that indicates the water or the liquid your water is composed to, which is leaving, so it's evaporating. The D is diffusivity, which is a coefficient of each of your solutes. So depending on what your formulation or colloids or whatever it is, they will have different numbers. So they're all studies, and they talk about how fast or how slow. So the speed of your molecules of solutes in that specific liquid, which most probably is water. So if you think about milk and dairy, that Peclet number will be calculated by knowing the kappa of water, because milk is a dairy compound and is composed mostly of water. And the D will be found for the micelles of fat in water. So there will be a specific number for a specific food. This number changes. Obviously, the kappa is dependent with temperature, and the D dependent is with density and viscosity. So it does change depending on different situations and also within the drying process. Now, we have different scenarios. If you have, as I mentioned already, if you have solutions, so as you see on the left-hand side in the blue square, where you have a solution is when you have solutes that has a diameter smaller than 1 nanometer. So it's not visible, it's not recognized, and it's completely dispersed into your water system or whatever liquid you have. And you can see that the Peclet number affect the particle formation. So when you have a high Peclet number, the molecules inside, they're very happy, they move fast, so they distribute first on the surface, forming an early shell and probably enclosing the water that is left in. When the Peclet number is low, so then the particles are slow, so they don't travel around, but the water is faster than the particle, which are inside there, so the water leaves faster. So usually in the scenario number 2, which is a low Peclet number, you usually tend to get small particles and solid, while in the high Peclet number, you have all of these weird shapes one. It's just because the shell is formed way earlier than the water evaporates. And the water entrapped into the formed shell can rather leave through the pores of the shells or burst the shells or create kind of an inflated ball. On the right-hand side, you have colloids and suspensions. So these are different systems. They have particles inside there larger than nanometers, and the definition are shown at the top. The Peclet number is the same process. When you have lower, you have mostly a solid particle, spheres, small. When you have high, you usually have a larger particle with voids inside. The theory here defines the number of 1 to be that milestones where you have larger, higher, or lower. Now, I have some example in the food world for low Peclet number, and they are desired. So companies is seeking for rather one or other aspects. Both are equally fine for different entities. For pharmaceutical world, as Bernadine mentioned, I did work in some pharmaceutical companies. They are more keen on having the low Peclet number because the particles are small and solid, so there is less spacing. So they can put more excipients within the capsules. So let's see the picture there for the oral delivery capsules. However, other properties are also higher strength and low crystallization chance. One example is lactase powder. They tend to crystallize, so they usually are amorphous, but when they have voids inside, they have more exposure to oxygen and can have oxidation. Another thing that can be redispersed is sometimes better, but most of the companies that works in dairy, they like to have something like that for spacing, so they can put more product within the same space. Another cool area that is now interested in spray drying and in particular for engineering is algae and microalgae. The technique of spray drying is the most common one used now to produce powder from microalgae. And they like to have something small as well, and with no spacing, the same reason for the cupazine for number 1. For the hyperclear number, there are those crumble shapes or open shapes. They are much better for dispersibility. Some companies that produce coffee or creams, because creams tend to be also sticky, so you want to put something else inside. Those shapes tend to have a much lower stickiness. Now, if you think about that crumble shape, it's way harder to collide one to another and to click and to make one agglomerate, rather the left-hand side is way easier. So in those cases, they like these shapes more, and they want to have powder shaped like that. Stickiness is a problem also for fruits. They create powders from fruits to use that as flavoring products, a source of flavors for yogurt, for other drinks, beverages, and so on. In that case, they want also those shapes that are really odd and unique. Infant formula, if any of you in the audience have children or know that, infant formula is a pain to usually disperse in water. You have to agitate or use hot water, shake it a lot. That's because the stickiness of lactose in there and components that are part of that are really hard to disperse, like whey protein. They like to have in these environments shapes that have water entering faster. So if you have the second shape here with a hole in there, the water contacts the whole structure and disperse better your powder into water. So the industry of infant formula is seeking to change and to improve better dispersibility by going into that direction. Those are some examples, and that is how you can influence, knowing the particle engineering theory, the shapes and the morphology and then the properties of the powder. These are just 2 examples, and that's the easiest one. There are way more, so you can dig more into the theory, but obviously for sake of time, these are the one I'm going to introduce to you right now. On the left-hand side, you have the influence of temperature. So in any droplets, even in the out environments, behave differently when we have a hot day or a cold day. So obviously the temperature, what does it do? It's enhance the evaporation of the liquid, so of droplets. So if you see on the left-hand side, you have for higher temperature, then the evaporation of water, it's faster, so the water leaves earlier. So we tend to have a larger droplets because of the water leaving fast and more particles on the surface first. So then you create a crumble shapes of the powder at the end. Then this is going to be depending a lot of your solids. That's why I'm saying this is just 2 simple scenario. Lots of solids will like to crystallize, like sugars and salts, which are major components in food. If they do crystallize, then the scenario does change drastically. You create crystals, nucleation can happen, and the crystal growth will affect completely your appearance and morphology of the powder. On the right-hand side, what you can do also is changing the concentration. So how much solute you have in your droplets. You can see from the left and the right, there is a big difference. So you have more components on the right. When you have more, obviously you have more solids and less water. So the water will be less to evaporate, and the solid will create first and earlier a shell. So the particle will be larger. So that's not usually welcome for some food products. You want to have a as small as possible particle of powder for spacing. When you have large drop particles, they don't like them because they tend to be sticky or also occupying a lot of space, unless they have crumble shapes, as I talked before. Now we go into some examples that I worked before in my career. I introduced a few, and then we go into what's going to happen here at UQ. What I mentioned already a lot, so what particle engineering can do is tuning what you have on the surface of powder. So you can see on the top left, if you have 2 components that are equally behaving, so they're equally crystallizing components or not, you can arrange by knowing the properties of those components to make one or the other to be at the surface. So in that specific case here, we have potassium and sodium chlorides. With sodium chloride, it's extremely common in most of the food, but potassium is a little bit similar to that, but the difference between them was the molecular weights. Usually larger molecular weights tend to distribute first on the surface, so they have a higher diffusion rates or diffusivity. You can change that by changing the temperature as shown before, or the concentration of component A versus component B. This is fundamental for a lot of companies to decrease the stickiness of what you have on the surface and that will affect the whole powder in the bulk properties. At the bottom of the left-hand side, you can also see crystalline materials like sodium chloride. That was actually the material that we used in there. You can see that by changing the concentration or the temperature, you can change completely the morphology of the particles. You can obtain a very teeny tiny particles in the range of few micrometers, completely full with no voids, until going to a very large particles with a lot of voids inside there. The case that was liked the most by the company we were working with, and in this case was AstraZeneca, was the middle one. The reason is that the shape of those crystals avoid and decrease a lot the adhesion forces, which are the forces that bond one or to another particles. By decreasing those forces, the powder are finer and they're not creating agglomerates. And for the pharmaceutical delivery, it's really fundamental not to create any whatsoever agglomerate because they will affect the delivery place and delivery system. On the right-hand side, we have also change of morphology as it happened already on the left-hand side. But the picture that you see at the top right is by using components that do not crystallize. In the specific here, there was cellulose acetate butyrate, which is a large polymers. That was a proof of concept, so showing that large molecules tend to have a big diffusivity and distribute on the surface earlier. So they create a thick shell. And in these shells, the water has to leave somewhere. And what that was doing, it was bursting on one spot and inflaming the whole particles, creating those lovely shapes that a lot of companies I mentioned earlier love. So that's showing that some companies are interested in using these polymers or other nature-based polymers that are all available to create this kind of lovely shapes that they want to have. On the bottom right is an example of a study that I've done with suspensions and colloids. We have cellulose nanocrystals and other metal oxide nanoparticles. The studies was aimed to create a powder for antimicrobial purposes. Metal oxide nanoparticles are usually highly antimicrobials, and when they place on some bacteria, they kill them right away. However, you need a lot of them to kill biofilms or a large amount of bacteria. Those amounts are not welcome, rather as pesticides or for the human delivery or for animal delivery. So in order to decrease the amount of metal oxide that you need to use, we create nanoparticles in microparticles to have a nice system to increase the contact area between the same amount of metal oxide nanoparticles and different bacteria. Another cool example that is part now of a product sold in Canada is the creation of a fat replacer from chia seed. This was a master student that could provide in the University of British Columbia. She was originally from Mexico and she was really interested in having some product from chia seed. So what she has done is compare different extraction procedures, and you see some on the top left. So we purchased chia seed from different suppliers from Mexico, and then we found the best way to get out the mucilage, which is a jelly substance from the seeds. And we dry the mucilage, store it there as an actually raw materials to use for further ingredients. So the best procedure that we found to be was the ultrasonic probe to extract the highest amount of mucilage from the same quantity of seeds. After that, we take the mucilage, we put it with other gums and sugar that I cannot disclose of what they are. We put them in the droplets, and we create this through particle engineering. These lovely particles, it's like a flower, and we use them as an alternative to fat replacer. So we do the sensory analysis to compare the common hazelnut chocolate spread like Nutella and ours. So we substitute fully the palm oil contained in Nutella with this powder. And you can see that A is Nutella and B is our product. Visually, people like our product because of the glossiness in it. So you can visually see from this picture as well how shiny the product is, and that's all from the mucilage from chia seed. So if you want to try it, you have to travel to Canada right now, and that's a picture on the left, on the very far right of the products when it's done. Another studies that I worked with was a spray drying of juice, of fruit juice. We work with strawberry, mango, and oranges. This also was a work of a master student, but she spent about a year to do that. The reason is that up in Canada, they produce a lot of berries. Strawberries and blueberries are the #1. and they don't sell all of them. So what they do is usually selling powders to X country where they buy the powder, redisperse into their products and sell it as fresh. So that's what it's done so far. So some people produce the powder by freeze drying and then milling or crushing. That's fine, but it's very sticky. So when you create powder like that, especially from strawberry, they create a huge amount of agglomerates, and it's very hard to disperse in water and create not pleasant final products for customers. So one way to decrease the stickiness is, as shown so far, is changing the properties powder. So in this case, we tried a few formulations. We use rather compare with the pures or ASP. Then we use maltose of different type with different DE ratios. We use maltodextrins, which is stands for MD. And then we use also HPMC, which is a cellulose polymer soluble in water. We also use whey protein. So the reason we use this material is because maltose and maltodextrins are already highly present in food and cheap. And then the other material, whey protein, is cool because it's coming from a source like dairy, so it's nature-based. And the other cellulose is because it was used before to encapsulate other compounds like minerals or different type of biotic compounds, and they're all FDA approved. The ratios were chosen out of a larger formulation matrix, so these were the best cases we had. So we went through with those cases to test them for further analysis. I just want to show you a few analyses, and here is a visual impacting pictures. Pictures are always great. That's what I always say to my student right now, to take pictures of every step they do, is because you can visually already tell which powder redispersed better in water. So all of these were taken with using the same amount of powder, same amount of water, and same amount of time of agitation. They were agitated for 5 seconds, so not a lot of times. So you can visually see that in the case of orange and mango, the whey protein that entrapped the juice, so this usually the whey protein is on the surface, it helped a lot the dispersion of the juice and decrease the stickiness a lot. So in those cases, the color was way more homogeneous. We did not test only the color, we did all the UV and other techniques to showing better the dispersibility. Now for strawberry, the whey protein did not really well work because in that case, the strawberry were not fully entrapped by the whey protein. Most of the reason is that the juice of strawberry is it was thicker and denser than mango and orange. So the amount of actual juice was higher in that case. What worked best was the polymer HPMC. So it has a larger molecules, so it can cover better the whole strawberry juice. And we had also better quantity of polyphenols and other contents of strawberry. I'm showing here again some of the stickiness values. So it's measured as force. We measure these with texture analyzer. A lot of you are probably familiar with that. So we put the same amount of powder on the probe, same amount of powder at the bottom, and I measure how much force it took for the powder to separate one to another. So it took higher forces, means they're stickier, so they're not good. Lower forces means they're better. Now the image is a little bit cut off, but the last point is for when using whey protein. So that's one of the best case for all of them, because the whey protein are repelling one to another and they're water repellents that help a lot in decreasing the stickiness of the powder. On the right-hand side is mold creations. So we have counted the mold count per hour, per area for all the products. So these are some cases. You see again, the lowest amount of molds is that when we have, in this case was a polymer, HPMC. The reason is that it's fully covering the whey protein. The whey covers is not fully enclosing. So it's a net of fibers. So it creates like a net all going around the juice. So there are some spacing for mold to get in contact with the fruits, while the HPMC is covering further and it doesn't dissolve fast. So it takes a while for the thick layer to be dispersed when exposed to room temperature and room conditions. And this was for after 1 month of storage. So we did not have a lot of mold growth in general in most of the cases, which was great. And that's what we wanted to do to increase the half-shelf life. Next examples. This is one that I'm very proud of because it's more humanitarian. And I think I talked before to some of you, but this was with the government of India. India specifically as a big issue for anemia, especially for women due to their dietary requirements for religious purposes. They don't eat meat, but they drink milk. So they don't get vitamin B12 a lot, especially and iron. So they want to have a fortified bread. So the picture you see at the bottom left is something that they worked on before. You can see the color, it's really not that welcome, and there's some clumps in there. So when you eat, you can feel some differences in the texture, which is definitely not pleasant. So what they wanted to do is create a naan bread, similar looking, similar color, all the same, just fortified, and they could give it to the populations, especially to more the poor one, to get some income of iron. You might say, why don't give them the supplements? It's because supplements are way more expensive to produce. And some people are not keen on getting, especially when you go in some villages, they don't like to get pills whatsoever. So having a product, a food product, fortified, is very important for some people. So what I've done there, we create a large formulation matrix, including iron and other minerals and vitamin B12 and folic acid. Vitamin B12 and folic acid are great supplements for pregnant women, which was the main target of these studies for anemic people. So we create this powder and then we test the delivery on the human body, but so far we reach only the animal test. You can see here that the cell top left has the highest absorption of iron when the powder contains both iron and vitamin B12 with a ratio of 50-50. The reason is that vitamin B12 boosts the absorption of iron naturally. So when we eat meat, we get both, and the combined action of both is fundamental for the optimal absorption of iron. So when you get supplemental iron, sometimes they are completely messed with what we eat. So if you eat dairy, for instance, zinc and iron counteract one to another. So we don't absorb any. So whenever you eat meat, don't eat yogurt or don't drink milk at the same time. Wait a few hours to consume any dairy. And that's also one of the problems in India they have. So then we have animal tests. We make the animals anemic. So we gave a diet that doesn't include any iron, which is the red one that you see at the bottom left. And then we divide them into 3 groups. One group have the control, so which is the common diet, the common pellets, the brown one that they eat every day. And then the second groups have the powder only with iron, so which is the green pellets you see. And the last group has the blue pellet, which has both iron and vitamin B12. Now you see on the bottom, on the top right, sorry, so that's the best case. So you see the different timing for the groups eating the blue diet have had the iron reaching the duodenum, which is where we want to go, in 40 minutes time, which is usually the digestion time of humans and also mammalian animals. And then after 90 times, it's pretty much everywhere in their feces. So it's already been digested and the amount is very low at that time. So it went to their bloodstream. Then blood analysis has been done different time steps. As you see at the bottom right, you have after 21 days, we make them anemics. That's usually the time it takes to make all the rats anemics. After that, that's where you see the 3 groups separating and having different trends. You see that the black, the square, so the square groups is the one that has a control. Then you have the circle is the one that has a green diet, and the triangle is the blue diet. So you see that the triangle is the one who recover faster in a couple of days, 3 days, their red blood cell count and their hemoglobin level reach almost what they had previous to the test. So that's way faster than what we have in the control, which usually takes 7 days to 2 weeks to get to the level you had before. So that was cool. Another aspect of this powder that it was white. So I forgot to mention earlier. Now, this is what I have and what I envision to do here at QQ. I want to continue working and analyzing the enhancing of powder properties. So continuing studies on properties of powder in general and particle engineering, as I've shown you so far. I want to continue working in encapsulation of bioactive compounds. I did not show anything I've worked before on the pharmaceutical aspects, but I want to continue working on that, especially for nasal, respiratory, and nose to brain delivery. Another part that I like to continue, and this is an example I've shown about the chia seed, is food fortification. So flour and other powder fortification, or biofilm, sustainable films, and also food processing. So improving some processing parts involving also powder, mostly powder of food. Like in this case, I put dairy or algae. Now I'm going to talk about some of the projects of the students I have upcoming. I did not mention all the master students. It is going to be a project of PhD students, so longer project, more thicker. These students will come from Norway. I received a scholarship joined from between the Q and ETU for her. She's going to work on seaweed and microalgae. Her project will be to create a fat replacer sourced by only seaweeds and microalgae. So there will be a first part where we extract here minerals and vitamins, and in DTU proteins and lipids, and then we'll combine the extracted bioactive compounds into one using the spray drying techniques to create powders that can be used for different applications and different products. There are some interest in marine bioproducts, the CRC, and other local companies that do like these projects and might be interested in doing some side projects on specifically on beauty products or microalgae for health. The other projects is the other PhD who is coming along this October. She's flying in a couple of days from now. She will work on creating again a fat replacer powder, so for food fortifications. What I envision doing is using some nut oils, macadamia, almond, and walnut. We still have to define which one to go. This will be the PhD goal, to do the first studies for review article, and using some techniques like cold plasma, which is already being used here in ECFAS, to remove the allergens from the oil. So to create an allergen-free oil, and then put the oil with other materials, which need to be selected to create a white powder with oil inside that do not leak and are stable in time, as a supplement for coffee whitener, so plant-based coffee whitener. And the students with their work in also drying coffee beans and coffee to create powders from coffee, and ultimately to create an instant plant-based cappuccino. So this is pretty much on my side. If you have any questions, I'll pass it over to Bernadine, who's going to show you how to answer to the questions. I think now is the time. If you need to ask, send that through the Q&A functions in Zoom.
Thank you very much, Alberto. That was a really fascinating presentation and a really great introduction to particle engineering, which was very accessible to me as a non-particle engineer. And such a great range of applications for this technology. Yeah. So we have one question to start with in the chat. Sorry, no questions in the chat. Questions only to go into the Q&A section. So we already have a question in the Q&A from Jackie from CNAFS. Thanking you for your presentation. Yeah, you know Jackie. Yeah. So she is wondering about the consumer study for the hazelnut spread. Besides appearance, did you also look at the texture in the mouth, and what about preference?
Yeah, so that's a good question. It was 2021, the end of 2021. So we were still on COVID time. Up in Canada, it was quite close. So it was very challenging. So what we had done is we prepare a box for each person who want to participate in the studies, send a box by mail to each of them who's going to go to their house. They open the box and they fill the questionnaire. Then they have to send back by mail the questionnaire to the department. So we reached about 100 people. The students spent about 2 months to prepare those lovely boxes. The people mostly focus on the look as a difference. The taste was very similar for all of them. We did not have any comments on the taste as a difference. So that was cool. Actually, we did not plan to bypass Nutella, which is the taste is amazing. But we were happy to have a better look, which was basically on the glossiness. And people like that, to have the shiny look on their bread, I guess.
That's how it's rated in my house too.
Yeah.
Jackie said thank you. So
you're welcome.
Uda has asked, may I know the source of iron you used for the fortification?
Yeah, sure. So we use iron gluconate, and that was the best one we found to be for the fortification purpose, and also the cheapest. So the government was happy with the cost, while there are other type of irons that are fancier but more expensive.
Um,
Thank you for the
I'll ask
question,
it.
Uda.
So they were the only two questions we have. I'd just like to ask a question about the chia mucilage that you're using in the Nutella. So does that mean that you have then increased also the fiber content of that?
Yeah,
So that's a
yeah.
pretty neat trick.
It is there are higher proteins and fibers in there, so that was a side effect of what we were looking for. We're mostly looking for reduction of the palm oil, which we were targeting as the main topics, but then we end up having more more things. So right now, the student is working in the startup companies, but she's very keen on selling it and marketing better. So that's part of the marketing also is the fiber content.
Can I ask one more question? I really like this.
Go for it. I'm here.
Just wondering about the accessibility to the equipment at UQ. Do you have all of this grand spray drying equipment and everything available?
Yeah, so we have right now we have a lab scale spray dryer and a large scale spray dryer. We have a 50-liter spray dryer. It is poorly used. It was mostly used by Dr. Bandari's production. So he has his cool products, which you might know, Perky. That was done here at the beginning. So they studied first in the lab scales, which he created his own device, very cool. Then we have other upcoming ones, spray dryer through FABA, through the Food and Beverage Accelerator. Then we have a new lab coming up here at PAX with 2 new food grade lab scales and 1 new non-food grade spray dryer. And I intend to purchase an electrostatic spray dryer, which is a little bit different and not completely different topics on the way. So that will take years from now.
In the meantime, we've got another point in the Q&A. So this is from Professor Mike Gidley. What do you see as the opportunity to design particles for specific delivery of nutrients to the large intestine?
That's cool.
Yeah.
Well, depends on which nutrients. So that's a very good question. So for the iron studies, for instance, we want to reach the duodenum, which is at the beginning of the intestine. And the main thing as it seems for the large is bypassing the acid environment in the stomach. So it's using materials that withstand the low pH. It's number one. So, and then having something that is not very water soluble because we are all composed of water. We want something that also stays for a specific amount of time, not disrupt. So we want to deliver the highest amount down in the system. So using specific materials like chitosan is a very popular one. However, it's very hard sometimes for the nutrients to be absorbed when you use chitosan because it doesn't want to open up at all. So some of them is going to stick on the chitosan. We actually use it for the iron to try it. It didn't work whatsoever, meaning that the iron was the liver not at all. So it was going to the feces even. So it was very interesting. So chitosan is very popular because of that, but it has some drawbacks. We end up selecting that cellulose materials because it's a large materials. It's not so soluble in water. It is. So we were able to work with that. And it was not opening up. The particles are enclosed in the pH low. So we test them for the release rate before doing any animal test. And then that work in the animal. We work also with insulin, and we use a mixture of chitosan and sodium alginate, and the insulin we also wanted to go to the intestine and the liver. So it's about combining different products and then other materials in there and being cheap for the purpose of not spending a lot of money when producing these powders. I don't know if I answered properly the question, Mike, but if you want to talk more, I'm happy to discuss personally with you.
I think I would very much recommend that. Alberto seems to have a great range of experience in this, and I think we could all benefit from talking to him more and following his research. So I'd like to close the Q&A there because it's about the right time. There's no more questions in the Q&A, and I'd just like to thank Alberto again for joining us today and sharing his
Thank
knowledge
you.
with us. Thank you.
Yeah,
So-
just pass to the next slide. Yeah.
So the next slide. So in 2 weeks' time, we will have the farewell seminar from Professor Nina Mitter, the Centre Director for the Centre of Horticultural Sciences. She will give a seminar on the borderless innovation, the changing face of horticulture. So we're all looking forward to hearing the seminar, but not to Nina, Professor Mitter leaving. So I'm sure we'll have a very good audience for that seminar. And so if you would like, sorry, the next slide. So if you would like to sign up, if you're not yet signed up for the alerts from the webinar series, there's a QR code there, or you can go to the website that's also listed there. And that's all the slides then, Alberto, I think.
Yeah,
so
that's all.
we'll just close it down. So
Oh.
thank you all for coming. Bye.
Thank you, Bernadine, for
Thanks.
helping out.